Okay, so welcome back to this next video in which we are discussing cutaneous nociceptors. Okay, right. So we've just discussed the 4 different categories of primary somatosensory neuron. Okay, and these are based on histological differences between the axons when you look at them in cross-section, basically. Okay, so if your primary somatosensory neuron has a massive great axon with a diameter of around 20 micrometers and is myelinated, then it will be categorized as an A alpha fiber. If your axon has a axon diameter of around 10 micrometers and is myelinated, it will be categorized as an A beta fiber. If your axon is around 5 micrometers and is myelinated, it will be categorized as A delta. And finally, if your axon is around 1 micrometer and is unmyelinated and is grouped in fascicles with a single Schwann cell looking after all of them, then it will be called a C fiber. Okay, right. We then discussed that generally nociceptors fall into one of these 3 categories, and you don't generally find nociceptors which are A alpha fibers. Generally, you have a huge number of C fibers, okay? Quite a few A delta fibers and hardly any A beta fibers, but you do find nociceptors which are A beta fibers. So really these ones are the main ones. You do have quite a few of these, and then this one, yeah, you do have it a bit. Okay, right. So basically what I want to now start with is differentiating the different nociceptors that you have of these 2 different types. So we'll group the A beta and the A delta nociceptors together as the A fibers, okay? And we will then discuss the C fibers and the A fibers. So basically, if you look at the nociceptors, which are C fibers, you can find that you can distinguish them you can separate them further into 4 separate categories. And this is based on what noxious stimuli will actually stimulate them to fire action potentials. So basically, let's start by distinguishing the C fiber nociceptors into these 4 different categories. Okay, so by far the most common C fiber nociceptors are what is known as the CMHC fibers. Okay, now what does this stand for? Well, basically M stands for mechanical. Okay, so that tells us that this C fiber will respond to a mechanical stimulus. H stands for heat, which tells us that this nociceptor will respond to heat stimuli, so either too hot or too cold. Okay, and the C stands for chemical, which tells us that the chemical mediators of tissue damage will also stimulate this C fiber. So basically, these C fibers are stimulated by absolutely all of the categories of noxious stimuli. Okay, so let me repeat that. This is a neuron, and it will be stimulated to fire an action potential not just by mechanical noxious stimuli, but also by the other 2, heat and chemical stimuli. So it's sensitive to all forms of noxious stimuli. Okay, now these have a more common name. They are called polymodal C fibers. Okay, poly meaning many, modal meaning form. Okay, so it will basically respond to many different stimuli is what this is saying. So polymodal C fibers. Okay, and these are the most common types of C fiber nociceptor that you find. C fiber nociceptors which respond to all forms of noxious stimuli. Okay, you then have the group of CMH. Okay, and you can now guess what this group is. This is the class of C fibers which will only respond to mechanical and thermal stimuli, but not to the chemical stimuli which signal tissue damage. Okay, so some of these nociceptors don't respond to all forms of noxious stimuli. They're more specific. Okay, then we have another form which is the CMC. Okay, and this then is the C fibers which will only respond to mechanical stimuli and chemical stimuli which signal tissue damage, but not thermal stimuli. So they lack the nociceptors, which remember are the receptors for the noxious stimuli that will detect heat stimuli, basically. Okay, right. And then finally, another type of C fiber nociceptor is known as the CMI-HI. Okay, and these are also called the silent C fiber nociceptors. Okay, now why are they called that? Well, basically, these C fibers will not usually respond to mechanical noxious stimuli or thermal noxious stimuli. However, if inflammation is currently occurring, and that's what this I stands for, if the area which has this silent fiber within it is currently undergoing inflammation, then inflammatory mediators will activate this silent fiber, and now that will arm this silent fiber, and it will now be capable of responding to mechanical noxious stimuli and heat noxious stimuli. So basically, these fibers don't usually work, but if the area is actually inflamed, okay, then they suddenly turn on and they will respond to noxious mechanical stimuli and noxious heat stimuli. Okay, so there are the 4 types of C fiber nociceptor, which we are now categorizing functionally. So we're splitting them into their different functions, basically. Okay, now we'll treat the A fibers, so the fibers which are myelinated, and they will either be A beta fibers or A delta fibers, but remember, this is just, kind of trivial. All that was different between them was different diameters, basically, and therefore different conduction velocities. Okay, remember that there's not some sort of God-given distinction between these, whereby this one has to be a nociceptor and this can't be. Okay, it's just they're categorized into that according to their diameters, basically. Okay, and nature isn't this discrete, it's much more continuous. There will be neurons in between, there will be neurons of 7 micrometers in diameter. It's not like all the neurons are either A beta or A delta, it's a continuous spectrum. We make these classifications to simplify things down for our minds. Okay, right, so let's now talk about the A fiber nociceptors. Okay, right. We can distinguish between A fibers and C fibers because there the distinguishing factor is less of a continuum. Does it have a myelin sheath or does it not? Then it's more binary, basically. Okay, right. So, just as we distinguished the different types of C fiber by their function, we're also going to distinguish different types of A fiber by their function. Okay, so basically you have AMH fibers, which are A fibers which respond both to mechanical noxious stimuli and thermal noxious stimuli. And you then have AM fibers, which are A fibers which just respond to mechanical noxious stimuli, and then finally AH fibers, which are A fibers which respond just to thermal noxious stimuli. Okay, right. So, the next thing I want to talk about is the fact that A fibers, okay, A fiber nociceptors are generally much more localized with regards to which portions of skin they innervate. Okay, so let me just draw you a little picture to illustrate this. So if we have our skin here, let's say this is the epidermis here, and this is the dermis, and we won't draw the lower down levels. Okay, so here comes our, let's say this is our A fiber. Basically, the A fiber might have a few little branches that innervate this portion here. Or it might have, lots of branches, but they all innervate this specific area. Whereas a C fiber might be more like this, basically. Okay, it might have branches that go off far and wide, basically. So, the receptive field, as it's called, which just refers to the little area of skin which the nociceptor innervates, okay? So this is called the receptive field. So in principle, what you could do is you could take a colouring pen and colour on your skin the receptive field of your neurons. That's what it means, a little patch of skin that will be innervated by a single nociceptor, okay? Basically, the receptive fields of C fibres are much larger than the receptive field of A fibres. So A fibres innervate a much more localized little patch of skin. Now, what's the significance of this? Well, there's more distinguishing factors between the A fibres and the C fibres that I firstly need to tell you about in order for us to understand the true significance of these receptive fields being different. So basically, the other thing I need to tell you about is that when we experience pain, let's say we just cut ourselves, okay, and we are now experiencing pain, initially you get a very, very sharp form of pain, okay? So when you've just done the damage, then you get a first form of pain called primary pain, which is generally very sharp and intense, okay? But that doesn't generally last long. Soon it's replaced by a different form of pain known as secondary pain, okay? And secondary pain isn't very sharp, it's very dull, and it sort of aches. It's the dull, aching pain that continues on for much longer, basically, okay? So the primary pain is generally because the A fibres are firing, basically, okay? And the secondary pain is when the C fibres are firing, okay? And the A fibres have now stopped firing, okay? So in primary pain, you'll have the A fibres and the C fibres firing, but then in secondary pain, you'll just have the C fibres firing. Now, when you have the A fibres firing, you get the very sharp pain, whereas when you just have the C fibres firing, you get this duller, aching pain. Now, what's this got to do with the receptive fields? Well, you might recognize the fact that the primary pain is generally very localized, okay? So this sharp pain, you generally know exactly where the problem is, okay? So even if you didn't know where you'd cut yourself, you would generally be able to understand, your brain would be able to picture the portion of skin that had been cut, okay? You'd know exactly where the damage was done. Whereas in the secondary pain, the pain's much more dispersed. It's not nearly as localized. If you closed your eyes and asked, where is this pain coming from? You might say your entire finger. Let's say you've cut your finger, okay? So you might say it's coming from your entire finger. So basically, the C fibres have much larger receptive fields, so when they are firing, you don't know where it's actually coming from. It could have been coming from over here, or it could have been coming from over here. All of those places are innervated by this C fibre, so you can't tell as well where the pain's coming from, whereas if this A fibre's firing, you know exactly where the painful stimulus is coming from. Okay, so that's the significance of the difference in size of the receptive fields of the A fibres and the C fibres. It means that you can locate exactly where the disturbance that is causing the activation of the A fibre is occurring, whereas in the C fibre it's much harder to tell where the pain is actually coming from. Okay, so you get much more dispersed pain, and that's why the dull secondary pain is much more dispersed, and the first pain, which is the sharp pain caused by the A fibre activation, is much more easy to pinpoint exactly where it's coming from. Okay, right, so that's the different types of nociceptor now. What I want to talk about now is how these nociceptors actually go into the spinal cord. So I want to talk about where their cell bodies are, I want to talk about where they go into the spinal cord, or the brainstem, because we're going to have to talk about the one exception. Okay, so let's now turn our attention to a bit more gross anatomy. Okay, right. So, firstly, the first thing to say is that most primary sensory neurons are going to go into the spinal cord. The exception is the primary sensory neurons which run in the trigeminal nerve, which are going to enter into the brainstem. Okay, so let's start off with a discussion of the spinal cord then. Okay, so basically, if we imagine chopping the spinal cord across, okay, so let's say this is the spinal cord here, viewed longitudinally at the moment, okay, what we're going to do is cut the spinal cord across and have a look at one of these sections, and we're going to cut it across at the perfect section where we will be able to see the dorsal and the ventral roots coming out. Okay, so let me draw now a cross section of the spinal cord here, like so. Okay, so you have the ventral sulcus right at the front, so I mustn't forget that. Okay, like so. Right, and then at the back you have a dorsal septum. So basically this is the spinal cord we're looking at in a cross section. Okay, and basically the spinal cord consists of white matter and then also grey matter. And the grey matter is clustered together in this sort of butterfly shape, like so. Okay, so all of this is grey matter in here. Okay, right. So basically what happens is if we have our sensory neuron, okay, and at the moment we're talking about a sensory neuron that comes in for a peripheral nerve that isn't the trigeminal nerve. We'll talk about the trigeminal nerve separately. Okay, so we're talking about sensory neurons that at the moment come in at the level of the spinal cord rather than at the level of the brainstem. Okay, so let's have the skin here, like so. So here's our epidermis, here's our dermis, and then we've got some cutaneous nerve here, like so. And basically, it then is going to make its way into some nerve, and then this nerve will go through all sorts of plexuses, and then finally this neuron will end up in some mixed spinal nerve, which is about to return to the spinal cord, okay, or emerge from the spinal cord. We are talking about sensory neurons, so we're talking about going into the spinal cord, but of course the whole point of the mixed spinal nerve is that it contains both sensory neurons and motor neurons. The sensory neurons will be coming back into the spinal cord, and the motor neurons will be leaving the spinal cord. So this is a mixed spinal nerve. And basically what happens is the mixed spinal nerve splits into two portions, like so, known as the ventral root here. So this is the front, this is the ventral direction, which is another word for anterior. And this is the dorsal portion of the spinal cord, which is another word for posterior. So this is your back, this is your front, your belly button will be over here. Okay, right. So this then is called the ventral root here, the root which is coming out of the spinal cord from the front, because of course this is going to join in here. So this is the ventral root of the spinal cord. And then the dorsal portion is the dorsal root of the spinal cord, and the dorsal root has a special structure, a special swelling known as the dorsal root ganglion here. Okay, right. So this whole structure, the whole thing here, this is the dorsal root of the spinal cord. Okay, and then the swelling of the dorsal root that is here, this is what's known as the dorsal root ganglion. And for short, the dorsal root ganglion is often abbreviated to the DRG. Okay, so the dorsal root ganglion, or the DRG for short. Right, okay, so basically in comes our sensory neuron into this mixed spinal nerve, and basically all the sensory neurons will go down the dorsal root. Okay, the ventral root is for motor neurons that are coming out of the spinal cord. Okay, all the sensory neurons go into the dorsal root. Okay, and then basically what will happen is their cell body will be in the dorsal root ganglion. Now, basically, these sensory neurons are all known as pseudo-unipolar neurons. Okay, and I want to explain what is meant by this fancy word, pseudo-unipolar. So basically, if you look at the structure of these primary somatosensory neurons, they have their cell body here. Okay, so this is their cell body with their nucleus in. And then they have an axon which emanates from the cell body, and it's a single axon. This is the unipolar portion. The fact that they have a single axon emanating from their cell body here. Okay, but then this single axon splits into two separate portions. It bifurcates. Okay, one of these is then called the peripheral axon. Okay, and this is the one which will innervate the epidermis. Okay, so this is the one that will go off to the skin. Okay, and I'll colour this one in green. And then it will have another axon which is going to continue on into the spinal cord, and we'll have this one in turquoise. And this one is called the central axon, okay, because it's going into the central nervous system. So, to highlight this on this picture, up here is our single axon emanating from the cell body of the sensory neuron, okay, and then here is the peripheral neuron, so I'll colour that one in green, and then here is the central, sorry, not the peripheral neuron, the peripheral axon, and here is the central axon, which is going into the spinal cord. Okay, right, so here is our primary somatosensory neuron, and the reason they're called pseudo-unipolar is really they do have two separate axons, but only a single axon comes off the cell body, so they're kind of half unipolar. They are, strictly speaking, unipolar because only a single axon comes off the cell body, but they're kind of frauds because they do really have two axons. Okay, right, so the peripheral axon is bringing the information in, it's then going to go down the central axon, and the central axon is going to enter the dorsal horn of the spinal cord. So basically, you can see that the grey matter is in this strange shape here, and we'll divide it into two. There's a central canal that runs down the centre there. Okay, so we'll divide the grey matter into two. Basically, these portions at the back, this process at the back, it's known as the dorsal horn of the spinal cord. So this is called the dorsal horn. And this horn that's at the front, this sort of extension of the grey matter forward, this is called the ventral horn of the spinal cord. So we have the dorsal and ventral horns of the spinal cord. Now, just to add a few extra labels onto the picture, there's little sulcus that you have at the front of the spinal cord here. This is known as the ventral sulcus. Okay, and this little fold that you have at the back here, this is known as the dorsal septum. Okay, so here we have the dorsal septum. Right, so those are just a little few landmarks that you should always put on your drawings when you're drawing the cross-section of the spinal cord. Okay, right. What I now want to discuss is the rexed lamina, because I want to show you which rexed lamina, or laminae rather, the central axon of the sensory neurons is going to go into. And at the moment we're keeping it fully general. This nociceptor here, it could be an A fibre or it could be a C fibre. They all do this. They all have their cell bodies within the dorsal root ganglia. Okay, so I hope that you're slightly impressed by this, because basically there is a massive grey axon running all the way from your skin all the way up to a little swelling, a little ganglion that lies next to your spinal cord. So you have these lying all the way up and down your spinal cord like so. Okay? And basically, it's one axon that carries it all the way from the skin to the dorsal root ganglion, so it's quite impressive, really. Okay, so we'll call it there for this video, and in the next video what we'll discuss is the rexed lamina, and we'll then discuss where the central process of this nociceptor is going to go to in the grey matter of the spinal cord.